Okay, so welcome everyone then to this 8th edition of the VPH Summer School. So this year is not called the VPH Summer School, it's called the Digital Human Twin Barcelona. And so because there has been change, the VPH Summer School was regularly then organized every year with Virtual Physiological Human Institute. And because the Congress of the Virtual Physiological Human Institute is happening every 2 years in September, so we have decided then to make the VPH Summer School so every 2 years, so the years when the Congress is not happening. So this year the Congress is happening. So 1 year we'll have the VPH Summer School then combined with the Digital Human Twin Barcelona. And then the other year, only the Digital Human Twin Barcelona that is combined with the Barcelona International Summer School. Just to contextualize the things. So we'll have then a full week talking about digital twin in health. So you will see that we have a series of lectures, so from Monday to Friday. So Monday we'll start then with Cristina, who I will introduce later on. So Cristina will give a talk that integrates biological data and with a clear clinical target. So that's a typical example of the complexity of the biology and the complexity to handle the biology to address health problems in patients that remain unaddressed. So be aware that the healthcare has progressed a lot in the last decades. And for example, infectious diseases have retroceded tremendously, but we still have a problem with what we call non-communicable diseases and disorders, which are the ones that you developed without being infected by anything, by bacteria or by virus. So some of them are linked with age, some others are linked with lifestyle, and here you can include cardiovascular disorders, you can include musculoskeletal disorders, you can include pulmonary disorders or respiratory disorders, and many more. So I'm just citing the most important ones. If you look over the last decade, the evolution of these non-communicable diseases and disorders, you see that while the burden of the infectious diseases has decreased worldwide, so the one of non-communicable diseases and disorders is actually increasing. Which means that despite all the knowledge, despite all the technologies that we are being developed, we are not able to cope efficiently with this kind of disease and disorders. So we will have then a series of talk that will illustrate then different aspects of the digital twin. And bear in mind that this is probably where the digital twin is actually most needed when we have to tackle with extremely then complex diseases and disorders, so multifactorial that we are even not able to identify a clear etiology or a clear pathophysiology. And very often we're talking about idiopathic. So idiopathic and medical language, it means that we have absolutely no clue from where it comes from. So sophisticated word to just say, I don't know, okay? So if you don't know where it comes from, then of course, the treatments you can provide are very limited to palliative treatment, so treating symptoms, and not treating the cause. So here, being able then to link different pieces of risk factors, which can be lifestyle, which can be mechanical, genetic, obviously, and then different biological interactions related with lifestyles, related with mechanic. So it's extremely important, and this is where computer model and simulations have probably their highest potential. So during this summer school, we will not particularly be talking about e-health or digital health in the sense that You have an artificial intelligence-based app that will tell you when you will die, you know? That will tell you whether you're having a good lifestyle or whether you need to do more sport. We're really talking about something that is much more sophisticated, probably much closer to research than to application, but we will see that there's still applications that are showing up on Friday. Okay, so of course, we're not talking about just one modeling and simulation technologies. We're talking about an entire continuum of modeling, simulation, and technologies. So we can map this continuum from data-driven models to really knowledge-driven models, knowledge and theory-driven models. So the most basic data-driven models that you can find are typically then statistical models, okay? So this is probably the oldest form of the data-driven model that we can find, which has then along the years evolved into machine learning as computer science, so has started then to reinterpret and then reshape the statistics. Behind machine learning and artificial intelligence, there are actually statistical methods behind, maybe not all of them, but behind a number of them, so they are. Okay, so then we would have then statistical models. So if we can generalize then statistical models, so we're coming then to meta-analysis. So meta-analysis, for example, has been on the forefront of the first effort to play with big data in health. So probably in the 90s and in the 20s, maybe it has started a bit earlier, but the real explosion was there. Unfortunately, it doesn't have totally fulfilled its promise, in the sense that, so we have started to play with different cohorts. So the first population cohorts have been created, so patient cohorts, population cohort, and then people were seeing that when they had a finding with one cohort, they were not able then to confirm this finding with another cohort. So really showing then the strong bias that you can have of the data-driven modeling and simulation, so depending on the sample, data samples that you have. So machine learning models have become more powerful, but they are not free of this bias either. So the good thing is that with the reemergence and the reinforcement and the generalization of machine learning and artificial intelligence, now much more attention is actually paid to data and to data quality, so which contributes then to make much stronger, much robust, and much more promising, so all this machine learning based on artificial intelligence. There's still one weakness of this type of modeling approach, which is necessary, but used alone, it still has one weakness, which is the mechanistic interpretability. So when the prediction works well, and then you have an answer to your problem, maybe you don't need to go further. But in the health domain, so it's very difficult that a predictor works 100%. So when it doesn't work 100%, so the medical doctors need to understand why. Because they need to have a clue about how then they should modify their interpretation and then maybe revise this model, okay? So this is where mechanistic modeling come into play. Mechanistic modeling has been then traditionally developed in the field of engineering. So first aeronautical engineering, civil engineering, and then it has generalized then to the health domain. There are some exceptions. You might be aware of Wolff's law, for example, in bone. There is also then the cardiac model by Huxley that are very early, the mechanistic models in the health domain. But really then the generalization that so comes from the engineering domain. So you have 0D models, you have 3D models, you have knowledge prediction models, etc. And so now we're coming to a point where then in order to address the need of data exploitation and then the need of interpretability, so data-driven modeling and mechanistic-based modeling so are starting then to come together, okay? And this is what is probably then needed then for the next generation of in silico medicine. So basically then all the modeling and simulation methods so come on a spectrum, and now it will become increasingly then difficult to just take a little part of this spectrum. So really one modeler in this century and in the next years, so someone who has to become increasingly holistic than in combining the different methods. So it might depend on the scales at which you're working. So the scales then leads to the degree of complexity. So lower down the scale you get, the larger is the complexity. So here you might need much more need of data-driven modeling. And we'll have examples of that. If you are very high scale too, when you're dealing with population-based modeling, so you have also an extremely high complexity. So here also you also need then the help of data-driven modeling. And when you're on very concrete and well-deterministic systems, such as the tissue and organ systems, or even the cell systems, so and then we will have an example here, so mechanistic modeling might work well. So you understand that when you couple the scales, you naturally have then the need to couple also modeling and simulation technologies. And then of course there are also these models and simulations, or these modelings that don't particularly request direct interpretability. And here we can talk about so all these new tools that are emerging, and actually these are probably then the closest nowadays then to application, which are then medical image analysis tools. So for example, if you have on a clinical medical imaging, so the medical doctor is struggling to see a tumor because of the resolution of the imaging, then there are machine learning artificial intelligence techniques that would help you then to generate high resolution images out of low resolution images done through the learning process with matching data, and so that the medical doctors then can see the tumor. So here you need to go then through the step of verification, to go through the step of validation, but at the end of the day, then the interpretative layer so is not that important. Okay, so then depending on your application, on the context of use, so then you always have a balance on up to which point interpretation is important, and up to which point so straightforward prediction is independent. And then it will also then help to balance into what kind of technology we'll have to combine. So as coming back then to the digital twin, so digital twin, if you look at the definition, it has 5 requirements, okay, which would be then connectivity. So connecting then the model and simulation, so among each other, among direct measurements. There is also a property of homogenization. So homogenization is then to be able then to generate data, then to pool data among then different individuals, and then to achieve, so you pass from personal-based prediction to group-based predictions to population-based prediction, and then so it obviously then helps a lot to address personalized medicine, in the sense that So if your personalized based interpretations are not easily interpretable because you're a particular case, okay, then probably if you've been able to homogenize the data and then check what happens in an entire population, then you can retrieve then the information that would help then to better understand your very particular case. This is this concept of homogenization. It should be reprogrammable, it should be smart, that's straightforward, and it should have the property of being able to leave digital traces. So leave digital traces, it means that you would have then a full history of your digital twin simulations that will help to know, medical doctors, in which context this person was in that way. No, in this new context, this person is that way. So you can then you can define a trajectory and then better understand how two persons that have started from the same point will evolve along then different trajectories and what has happened. And then again, combined with homogenization, you can better understand what is happening, and then you can obviously also better understand different complex pathophysiologies that would be extremely highly multifactorial. And then finally, it has the properties of modularity. So modularity is, I want to explore the brain, okay? And maybe if I want to explore the brain, I need to explore then the cardiac system. And then I need to be able then to couple these two systems, okay? So that's an example of modularity. We can cite thousands of examples, okay? But this is a typical one, okay? So you might not need to have an entire simulation of an entire person if you're modular, because some systems are strongly coupled on the short term. Some other systems are strongly coupled, but only on the long term, okay? So then this modularity also allows to be more efficient in this digital twin technology. So along the VPH Schirmer School, we will see actually, the days are a bit fractioned in terms of modularity. So unfortunately, we won't have an example of an entirely fulfilled digital twin because it doesn't exist yet in health, of course. And but we will see extremely nice examples already of how mechanistic modeling, data-driven modeling are connected, and the different models. So models then to integrate data from outside then into models, then this will happen tomorrow. Modules of modeling at different scales, which will happen on Wednesday, and then all the problems of implementation and then validation of the different models, which will happen on Thursday. And then on Friday, you will have an extremely nice example, so mostly in-house technologies, but we will also have a talk from a company that has been co-founded by a researcher from IBEC. So about what is already readily then exploitable and what is the added value, and what are the steps in this very specific field of digital twin, what are the steps that are important in order then to push forward these technologies into an asset for the population, so going through entrepreneurship. Okay, so I'm finished with the introduction. Just bear that in mind then. So during your hands-on, okay, try to always contextualize then your hands-on in this overall picture of the digital twin. On Friday, there will be then the hands-on presentation in the afternoon with an independent jury. It's a competition, there is a prize. And then you will be evaluated then by an independent jury. So you basically then will have to convince them that you're not always good at talking and explaining things, but that you're able then to properly position then the technology and what you've done during the week, okay? So I will send you this afternoon already, I will send you then all the instructors and the different criteria so that you can be guided and upfront in this positioning exercise. So I'm done with this introduction. Has anyone a question about the program? I'm not showing the program. I guess everyone has it. Yeah? Okay, perfect. For this afternoon, everything is under control. You know where to go for your hands-ons. You know who you are going to do your hands-ons with. Yeah? Who has no clue? Fantastic. Okay, so now then it's then time to introduce Cristina. So we're very lucky. So Cristina then is coming from the other side of Spain, so on the west, and so she has a PhD in pharmacy, and she's a researcher in the Galician Health Service, SERGAS, at the Hospital Universitario de A Coruña in Spain. And she is leading the proteomics unit of the Rheumatology Research Therapy Group at the Network Research Center of Bioengineering, Biomaterials and Nanomedicine of the Spanish National Research Institute of Health. She is the scientific director of the proteomics unit of ICTS. I don't know what it is, but you will be able to explain that probably. Nanobiosis. And so she's an expert on an application of omics strategy, so to study pathophysiology of rheumatic disease. So I guess you'll be mostly talking about osteoarthritis.
Yes.
And so then it's my great pleasure now to leave you in the hands of Cristina, who will explain her research. Thanks, Cristina.
Thank you. Thank you, Jérôme. I'm going. I have
my
my microphone.
Thank you.
Let's put the presentation here.
For the discussion, so we have an extremely comfortable time for discussion. So again, just get inspired, make questions, okay? Because there are people online, I pass the phone for the question, okay? Don't ask question if you don't have the phone. And for people who are online, I don't know, well, we'll ask you if you have
3 people online. So, good morning, everyone. Thank you for Jérôme for the nice presentation. So, as Jérôme said, I'm Cristina Ruiz. I come from La Coruña. I'm not such as good a speaker as Jérôme, so I hope I can translate and share with you the objective of my talk, that it's rather clinical, so I'm really not aware of your background, so I hope I can just make you follow this talk, that I'm going to focus in osteoarthritis, and also I want to emphasize also the value that proteomics, that is the specialty that I work in, has to answer clinical questions and answer problems in biology. As Jérôme said, I work in a hospital in La Coruña and in a rheumatology research group. And so in this talk, this is the outline that I will follow. I first will want to introduce you a little bit in the disease that I will talk about, that is osteoarthritis. Then I will have, in a second part, I will talk more about precision or personalized medicine and why is it important in osteoarthritis and in general in all diseases. So what I will talk here is applicable to other diseases and how proteomics and other omics technologies can be useful for this. And then in the final part of my talk, I will show you some application of proteomics that we have been doing. I have been working in research, in this kind of research for like 20 years, and things have evolved a lot during the last decade, so it's really something that in a couple of years gets old, so there are many things moving there, both for the discovery of novel biomarker candidates and also to develop predictive models based on traditional statistics. That is something that are the the approaches, the strategies that we have followed more usually, but also with machine learning and other kinds of data analysis. And this is also very important for precision medicine. So I will begin with showing you a little bit what osteoarthritis is. This is the most common form of arthritis, and what does this mean? This means pain in the joints. So it's characterized by pain and also structural damage in the joint. It affects millions of people worldwide, and it has an increasing global burden because there are no effective treatments. So this is an important clinical unmet need. And also it's related with age. So with the aging of the population, it's expected this is expected to grow even more in the next decades. So in OA, traditionally, this disease, this is a knee joint, for example. Traditionally, it has been considered like a passive process that is caused as a result of the mechanical wear of cartilage. Cartilage is this tissue that is a thin tissue that it's covering the end of the bones in the joints. So it lubricates, it softens so that there is no physical damage. The bones are not one to each other. And it was considered previously like a degenerative disease. But currently, this concept has changed and has evolved to a more modern one, in which osteoarthritis is considered really an active process that involves not only the cartilage, but also all joint tissues, the cartilaginous cartilage, the meniscus, the synovial membrane, that is this thin layer that is covering the joint capsule, the ligaments, the subchondral bone, etc. And it's also not considered a degenerative disease, but it has this very big definition that was released by the society that is leading the research in osteoarthritis worldwide. So it's involving movable joints, and it's characterized by cell stress and extracellular matrix degradation, but it's also an inflammatory disease. That it was previously not considered inflammatory, but nowadays it can be considered also inflammatory disease. And also what is important is that it's shown that this disease manifests first as a molecular derangement. So molecular changes are the ones that start the disease, and then this is followed by other anatomic and physiologic derangement, not only in cartilage, but also in bone, as I told you earlier, joint inflammation, loss of normal joint function. So this is like the concept of osteoarthritis that we have nowadays. Regarding the prevalence, I told you that it's the most frequent rheumatic pathology. So this means that worldwide there are more than 650 million people suffering this disease, about 40 years old. In Spain, 1 million. So this means this is a very important problem, health problem. This is the last study that was led by the Spanish Rheumatology Society, that's called EPISER. Quantify the total osteoarthritis cases in around 30% of people above 40 years. And most of the joint that is most frequently affected is knee OA, followed by hand and by hip. So the osteoarthritis can have different localizations. In this talk, I will focus more on knee because it's the one that is more frequent. But it's also more frequent in women than in men. And 1 million, it's estimated on 1 million in Spain, 1 million people in Spain is suffering this disease. And it has really a huge socioeconomic impact, estimating it in about 0.5% of the gross domestic product, GDP. And this socioeconomic impact can be related with direct costs. Mainly, these direct costs are related with surgery, because when the disease is in very advanced stages, the only solution is joint replacement by a prosthesis, but also treatments and long-term care. There are also indirect costs related with the decrease in productivity because of work absenteeism, and it's not a mortal disease, but the disability that it causes, it's also a cause of premature death. And also there are a lot of intangible costs because it essentially causes a loss, a very important loss of quality of life and limited activity to the people that are suffering this disease. There are a lot of, this is a very complex disease, multifactorial. So there are several risk factors that can be identified in osteoarthritis. Some of them are non-modifiable, such as gender. As I told you before, it's more frequent in women than in men, or the race or age. So these are the most important risk factors. And other risk factors are modifiable, such as diet, physical inactivity, lifestyle, etc. So there are some things that we can intervene and we can modify, but others are not modifiable. To diagnose OA, as I told you before, it's characterized by pain and also by structural damage. Regarding pain, that is the main symptom of OA. We have these pain scales that are evaluated by the physician. The physician makes like questionnaires to the patient, and due to the collecting the responses of the patient, you can do like a scale of the pain that the patient is suffering. That this is, of course, subjective. So these are subjective measurements. And then we can also observe the structural damage that is taking part in the joint. And to evaluate this structural damage, the reference technique is X-ray. These are examples of X-rays of the knee joint. And in this case, the degree and of the severity of the disease can be scaled with scales that are based on these X-ray images. This is an example of the Kellgren and Lawrence scale, or KL. This is a radiography of a patient with a healthy patient, healthy individual, with a KL of 0, where you can see the space that is between the tibia and the femur. And here you can see that as long as the disease progresses, this space is narrowing, and then it leads to a total destruction of the joint, and then you end with a prosthesis. The problem with radiography is that it's quite insensitive. When you start seeing that these changes at the level of radiography, it's known that the patient has lost already like 10% of the cartilage, and also the cartilage is really not detected in the radiography. So this is an indirect vision of what is occurring in the joint. So the magnetic resonance is much more sensitive. With resonance, you can see many more things, but it's really difficult to implement in clinical practice because, as you see, there are many people suffering osteoarthritis, and you cannot do MRI to each patient in each joint because it would be very, very expensive. So we have the limitations essentially for the early diagnosis of the disease. And also what is very important, we have a limitation for early diagnosis and we have limitations for therapy because as I told you before, this disease has not effective treatment available. So what the physicians do is recommend non-pharmacologic treatments that aim to act on modifiable risk factors, such as, for example, weight loss. A very important risk factor for knee osteoarthritis is the weight. Or moderate exercise, or trying to strengthen the muscles and protect the joints, etc. Or there are people that have a malalignment of the joint, so you can correct this. This is something that you can act on. And then there is also pharmacologic treatment, but it's always controversial because it does not stop the disease or reverse the progression, but only act on symptoms such as pain, for example. So we have analgesics, we have non-steroid anti-inflammatory drugs, we have corticoids also, and we have this group of drugs that is the acronym for symptomatic slow-acting drugs for osteoarthritis that are chondroitin sulfate or glucosamine, that maybe some of you have heard about it. There are people that say that they don't work at all, but for many people, maybe it's in osteoarthritis for the clinical trials, we have a very important impact of the placebo effect. For the pain, because people start taking things such as chondroitin sulfate and they feel better, but you don't know if it is really having an effect on the pain or that is just the impression of the patient. So in the case of OA therapy, clearly we have to move into more personalized and precision medicine strategies because we have here a super important unmet clinical need. And to move to precision medicine, most of you know that we are now more or less here in an evidence-based medicine, that we are We collect data from clinical trials in which drugs are tested, and we recognize patterns by statistical methods, and we try to manage the patients in base of this pattern recognition. So what we are moving now, this is future, but it's present, we are moving now here, is into the use of algorithms, into the use of massive data, that enables the physician to make decisions based not in predictions but on knowledge. And in this case, the precision medicine is when the prevention, diagnosis, and the treatment are adapted to the molecular uniqueness of the patient and its disease. So to shape the management of your disease to the molecular uniqueness of your patient and of your disease, you, of course, need to better define the diseases, especially at the molecular level. So you need to stratify the patients, you need to better characterize the patients molecularly, and in this case, you will be able to give the right patients the right drug at the right moment. So, and this is where we are moving now. So in the case of osteoarthritis, we can stratify the patients clinically by observable clusters of features that are called the phenotypes. And there are 4 phenotypes that have been, in the recent years, have been characterized or identified. The mechanical phenotype that is more associated with, for example, sport professionals, so people that are that play sports very intensively when they are young. If they have a meniscal tear or they break their ligaments, so they have, this is an important risk factor for osteoarthritis, that will have develop osteoarthritis early in time. Then there is this aging phenotype, that is the traditional view of osteoarthritis, that is when old people will have osteoarthritis, old people will have pain in their joints, and this is life, so you cannot do anything about it. Then there is this metabolic phenotype that is associated with diet and with lipids and with fat diet, alteration in metabolism, and also the inflammatory phenotype, that is when the joint is really characterized by a really hard inflammatory process that is not common in osteoarthritis, but it can be. So we have these different phenotypes, and when we go to the molecular basis of this is when we start looking at the OA endotypes. In this case, the endotypes are the molecular mechanisms that are distinct and are biologically linked to these clinical features. So we go to the molecular signatures that underlie these phenotypes. In the case of the mechanical phenotype, we see a lot of matrix degradation, of loss of collagens or proteoglycans of the extracellular matrix of the tissues that are in the joint. In the case of the aging phenotype, we look at processes like as chondrosenescence, how the cells in the joint are going senescent, or apoptosis processes. In the metabolic phenotype, we looked at altered metabolism or mitochondrial dysfunction. That can be the ground of the phenotype that we are seeing. And in the case of the inflammatory phenotype, the presence of inflammatory mediators, cytokines, imbalance of redox processes, etc. And this is important because the OA pathophysiology, this is the joint, has these key events that we can see that are taking part in the joint: apoptosis, an increase in apoptosis, an increase in senescence, a decrease in autophagy, that is limited clearance of damaged cells. We see these alterations not only in cartilage, but also in the synovial membrane, in the bone marrow, or in the subchondral bone, that is the bone at the end of here in the joints, and also at the level of the cells. In the synoviocytes, that are the cells present in the synovium, mesenchymal stem cells that are in the bone marrow and can be programmed or can be differentiated to repair the joint. In the cells of the subchondral bone that are osteoclasts and osteoblasts, and also in the chondrocytes that are the cells present in the cartilage. And by proteomics, we have analyzed all these kinds of cells of tissues, trying to find which pathways can be altered and can end into different osteoarthritis phenotypes. It's also known that not only we can look at this, but we can look also at the molecular alterations that are taking part here. So in osteoarthritis process, there is an increase of metalloproteases and agglutinases that are enzymes, proteases that break the proteins of the extracellular matrix and leading to their destruction. There are also inflammatory lipid mediators, such as prostaglandins, There are free radicals, such as nitric oxide, that is known that the presence of nitric oxide increases the expression of metalloproteases and increases the cell death. And when you reduce the synthesis of nitric oxide, you can reduce the cartilage degradation. And also the presence of cytokines, pro-inflammatory cytokines that is known that participates in the OE process, such as interleukin-1 beta or TNF-alpha. So this is, at the molecular level, you can see lots of processes that are taking part in the disease and that are interesting, because then, regarding of these phenotypes and endotypes, then you can also classify the osteoarthritis into what is called different theratypes. That is how are you going to manage the treatment of these different phenotypes. So in the case of the mechanical phenotype, you are looking at a lot of extracellular matrix destruction. So the management solutions could be surgery to repair the alterations that were caused mechanically, or trying to implement regenerative medicine strategies. In the case of the aging phenotype, you can use senolytics or xenomorphic drugs to stop this chondrosenescence. In the metabolic phenotype, you can focus on diet or anti-diabetic agents. And in fact, there are some anti-diabetic drugs that are used for weight loss that are very popular now, the GLP drugs, that are showing good results to stop the metabolic phenotype in osteoarthritis, and also anti-inflammatory drugs in the case of the inflammatory phenotype. So this is something that has evolved from the last year. So in this case, it's clear that we need biomarkers, because these markers will help us to define in a better way the diseases at the molecular level, and to find these biomarkers that can be used to implement its precision medicine strategies. It's a good idea to introduce omics technologies more deeply into the procedures, because omics technologies will allow us to better characterize biomarkers and to enable us to establish precision medicine strategies. This is a scheme of the different omics technologies, from genomics, transcriptomics, proteomics, and metabolomics, then go to explain the genotype. The genomics explain the genotype, and they go they move to the phenotype. The phenotype is what we observed, and we observed it by molecular markers and also by clinical features and imaging markers. And all of this is what builds the phenotype. But what is important is that the molecular markers move here all through this process, and they are characterizing the endotypes, and this is what, in our case, proteomics, it's in between all the omics technologies, is the one that gives more functional meaning to what you are observing, because proteomics, it defines proteins, and the proteins are the molecules that are doing the function in the cells. So it's not that the other omics are not important, but when you want to translate into a functional meaning what you are looking, the proteins are the ones that give you the key and the answer for that. So that's why we study proteomics. So this is another way of showing how the timeline, how osteoarthritis progress in time. So as I told you in the definition, that it starts with molecular derangement that take place more or less 20, 25 years old. There start some molecular changes that are not observable, so you cannot see. This is what can be called very early osteoarthritis, that in principle is undetectable, and you have no symptoms, you have no imaging features. Then you move to early osteoarthritis, in which you can start looking at incipient features at the level of resonance, but still the X-ray are negative, and the person has not symptoms at all. And after like 25 years, from 25 till 50 years old, when you are 50 years old is when you can be diagnosed of osteoarthritis, really, because you have a consolidated osteoarthritis in which you have symptoms, you have MRI features, and you have also a positive X-ray radiography showing alterations in the joint. So this is consolidated, but you have lost 25 years of progress of the disease. And after another 25 years old, more or less, you will end with a prosthesis. So this is something that cannot be, you cannot do anything about it at the moment, but it's where molecular biomarkers have an importance here, because they start, this molecular rearrangement start here, and very early stages of the disease, you can detect them, and they can help to predict how the patient is going to evolve, and help the physician to guide precision medicine strategies to try to stop or to try to slow the progression of the disease. So molecular biomarkers have been studied for a long time. There are several protein biomarkers that have been described in OA. They are related with the different cells that are participate in the processes in the joint. Many of them are related to matrix destruction. Some of them with signal inflammation, synovial inflammation, and others are proteases. So there are many of these have been studied. They have been introduced in clinical trials. They have been introduced in meta-analysis in the last decade. But really none of them is really now implemented in clinical practice. So the one that has shown more potential is CTX2. That CTX2 is an neoepitope of type 2 collagen. Type 2 collagen is the main protein that is present in the extracellular matrix of cartilage. And in this case, the biomarker is not the type 2 collagen, the entire type 2 collagen, but neoepitopes that are released by the action of proteolytic enzymes. So it is known that the enzymes are damaging and degrading the cartilage, and they release these neoepitopes that can be good biomarkers of the disease. So these are the ones that have been more extensively studied since 2012. Other extracellular matrix proteins such as aggrecan have also some neoepitopes that have been analyzed as biomarkers, and markers related with collagen degradation, with collagen system, with oxidative stress, matrix turnover, and other several molecules, but none of them has been introduced in clinical practice. So we have really a need for novel proteins that can be useful to be inserted in models that can help the guidance of precision medicine strategies. So here is where proteomics has a value. This is a traditional biomarker pipeline where you have a discovery phase in which you screen many proteins or many analytes in this case in different types of samples. And by this discovery phase, you can search for biomarker candidates. Once you have a list of biomarker candidates is when you can develop an assay that can be translated into clinical practice, can be implemented in clinical practice, and validate this biomarker. And when you have these validated biomarkers is when you qualify it for clinical use, and you can launch it into a new product that can be commercialized and included in clinical practice. So this seems easy, but it's really very complicated. We have a really huge bottleneck here, because very few biomarkers reach this stage to be implemented in the clinical use. And proteomics in this case will help here, because as an omic strategy, it enables the screening, the large-scale analysis of thousands of proteins at the same time. So if we want to discover new novel OA candidates and verify it, then it's when we can use proteomics. And there are two different strategies for biomarker search. The first one would be shotgun proteomics, in which this is an hypothesis-free strategy. We don't know what we are looking for. What we want is to identify the highest number of proteins possible in your sample. So, and to this end, we use mass spectrometry. Either there are different types of acquisition for mass spectrometry, but using mass spectrometry is where we have a sample of tissue sample, or a cell sample, or a serum or biological fluid sample, and we look at the proteins that are present there. And the second strategy would be, this is very useful for discovery proteomics. And in the case that we have a list of candidates, it's when we move to targeted proteomics. And in this case, we analyze, this is hypothesis-driven. We analyze a specific panel of proteins. And the most used strategy to do this are immunoassays. So we know the list of proteins that we want to look to. We have antibodies against these proteins, and using either ELISAs or traditional immunoassays, or this kind of multiplex technologies that are based in magnetic microspheres that are linked to an antibody, we can detect the proteins and quantify them in the samples. So based on these different strategies, I'm going to show you here an example of a work that we carried out several years ago, like 10 years ago. But it's an example of discovery of biomarkers in an in vitro model. And in this case, we have samples. These are femoral heads from patients. And you see this is a healthy femoral head, and this is an osteoarthritic one. I don't know if you see the lesions here, but this is the surface of this femoral head is really damaged. And we took explants, cylinders from the cartilage here, from the normal, the healthy cartilage that is here. And then in the case of the osteoarthritic samples, we took a cylinder from the wounded zone. This is the one that is very lesioned, where the damage is. And then to the zone that is adjacent to the lesion. So this is the unwounded zone and the wounded zone. So in this case, we would see molecular changes that are taking part in the joint but are not macroscopically visible, you know? So you can see here, this is an histological assessment of the proteoglycans. As you can see here in the normal, in the healthy cartilage, you see this red stain is related with the amount of proteoglycans. So this is a healthy tissue. And when you see the lesion, the proteoglycans are really depleted there. So you can see it here. So we put these cartilage explants into microwells with culture, and we put them in culture to let the explants release proteins to the medium. And in that case, we would see the proteins that are released from the tissue that in an in vitro model can be potentially good biomarkers of what the process that is happening in the tissue. So we took what is called the secretome, that are the proteins that are released to the medium. And in this case, we used a labeled proteomic strategy in which we labeled the conditions, and then I'm not going to go in deep into this, that is very like experimental details, but we can quantify these proteins, and we see different profiles of release. There are proteins that are in the healthy joint when the lesion starts in the unwounded zone are already differentially released, so the release is increased, and then in the wounded zone are more released again, even more released. Others that are the highest release is in the zone adjacent to the lesion, but once that the lesion and the tissue is damaged, they stop being released, so that they could be markers of early disease but not of advanced disease, and others whose release decreased with the damage. So we can see these different profiles, and we can see how these proteins are related with extracellular matrix organization, with stress responses, or immune and inflammatory responses. And we can look at different markers, different proteins that can be potential markers of the disease. And in that work, what we finally did is we analyzed one of these proteins that we saw that has already an increased release to the medium in the unwounded zone of the cartilage is the periostin, that is a protein that is related with the subchondral bone. And we saw in synovial fluid that indeed this protein is increased in synovial fluid from OA patients compared to non-OA patients. So this is something that can be detectable at the level of the synovial fluid that it's in the joint at the level. So this is an example of what we look at when we look at proteins, but in this case we also, using this in vitro model, we also looked at neoepitopes, so proteins, endogenous peptides, not the whole protein, but peptides that are released by the participation of metalloproteases or proteases. And what we did is using the same in vitro model that we used previously. In this case, what we did is we filtered the secretome to enrich this sample enrich it of endogenous peptides, filtrating and analyzing those molecules that are smaller than 10 kilodaltons, that are small peptides that are released there. And we identify them by mass spectrometry, and we found more than 1,000 different peptides that are released from the cartilage. From this list of more than 1,000 peptides, we select some. These peptides belong to 100 different proteins. We select some candidates, 55, 50, 54 peptides for quantification. We developed a targeted proteomic assay, and then we verify and validate these differential peptides in independent samples of patients. And finally, this led to the identification of 8 peptides that have marker value of articular cartilage turnover in osteoarthritis. This is an example of what we found. This is one peptide from the prolargine that is also an extracellular matrix protein that is present in the cartilage. And these letters correspond to the amino acids of the peptide. And as you can see here, this peptide is secreted. It's released the highest in knee osteoarthritis. This is knee when it's marked with a K. And this is hip, because we analyze hip cartilage and knee cartilage. And this is the highest release is detected in the wounded zones of knee articular cartilage. And you can see here how this release is statistically significant. And with this information, we can build these rock curves that I don't know if you are used to look at these curves, that are a graphical way of looking at the classification performance of the model that is built with this peptide. So looking at this peptide has, I don't see it, like around 0.60% accuracy to classify the wounded cartilage between wounded cartilage and healthy ones. So you can see here how you can make this kind of models using this the abundance of these peptides in your samples. But of course, in vitro assays then are difficult to be translatable into clinical practice, because you are seeing this in culture, in cartilage explants, and what we found is that then it's very difficult to detect these peptides. We try to detect this prolargine peptide in serum, for example, or in synovial fluid, and we fail to do that because they are released at very small amounts. They are enriched in this type of samples, but if we want to look at them in serum, for example, you are not able to detect that. So we move to analyzing directly serum from patients. We have performed proteomic analysis in synovial fluid and also in serum. And the advantage of working with serum is that you have thousands of samples in repositories, because the serum is the sample that it's most commonly collected from patients in the biobanks. So we have a lot of samples to do proteomics analysis and biomarker studies in more large-scale cohorts. Doing proteomic analysis of serum, we wanted to try to directly discover and validate in a biological fluid that is easily accessible. So this would have a easily translatable application into clinical practice to study circulating proteins that could be useful to improve the diagnosis and prognosis of osteoarthritis. And to this end, we have used different cohorts that we have in our lab. One is the ProCoag, that is a cohort that was developed in our group from osteoarthritis patients. Then we also analyzed this cohort that is called OAI, that is from the NIH in the US. And also this cohort that is called the Approach, that it's a cohort that is built within an European project. And the clinical centers that collected and recruited the patients from this cohort belong to 4 different European countries. So there are different cohorts in which we could do our biomarker studies. And I show you here the timeline of osteoarthritis and how molecular markers are in all through the timeline of the osteoarthritis progression. And the reasoning to do these studies is to then use these biomarkers and these proteins that we see altered in serum to build predictive models that can be useful for precision medicine. So if we look at molecular markers here at the beginning of the disease, in the preclinical phase, and also when the radiography is still negative, so this is not really a diagnosed disease, we can predict the incidence of the disease. While if we look at the biomarkers, at the modulation of the biomarkers in this time, once the disease is radiographically, the joint is radiographically damaged, we can predict the progression of the disease in time. We can also predict if the patient is going to have a total joint replacement at the advanced stages of the disease, and we can all throughout also this timeline predict if the patient is going to respond to a treatment, a specific treatment or not. So these are different stages in which we can do this predictive modeling. So I will start with the prediction of progression. We have done this study in the approach cohort. The approach cohort is based on this approach project that I told you before, that it's an initiative from the Innovative Medicine Initiative from the EU. And in this project, we participate 25 European and 1 US partners. And this includes not only academic partners, but also small enterprises and big pharmas. So, for example, GlaxoSmithKline and two big pharmas, or 3, were participating in this project. And this project included the development of this cohort. That is a cohort that was followed for 2 years. And in this cohort, what the aim of this project was to try to recruit, to improve the results of the clinical trials, because as I told you before, the osteoarthritis doesn't have any drug that is effective, and this is because the clinical trials are failing one after each other, so they have not succeed at all. So what they wanted to do is to try to identify those patients that will be more likely to progress in the disease, because those are ideally the best to be recruited in the clinical trial, because they are going to progress, and you can see if the drug is going to be effective or not. Because if you do a clinical trial during only 2 years, many of your patients will not progress, and you are not going to see anything if the drug is effective or not. So they recruit almost 300 patients. And then at the end, they observe the different phenotypes. If the patient has progressed in pain, that would be the group P. If the patient has progressed in structure, the group S, or if they have not progressed at all. And by the proteomics, we would like to look at the molecular endotypes, at the proteins, how the proteins are modulated here. We would like to see if are there any proteins that are different between the 3 groups at baseline that would be good predictors of the progression, either in pain or in structure of the disease. If we look at the end of the follow-up, we would look at if there are any proteins that are different at the end once the patient has progressed. That would be markers of the progressions. And also, we looked at the difference between the baseline at the end. So if we could look at the dynamics and how these proteins are modified during the progression. So we looked at this. And to do that, we did this approach. We select some patients, 45 patients at baseline and 45 patients at the end of the study. We analyzed their proteins in the serum of these patients. We looked at some candidates, and we tried to verify them by Luminex. I will show you some of the results that we had here. And in this case, we are now finalizing the data analysis of the validation, and we have found 3 proteins that seem to be good predictors at baseline of the progression of pain and structure. So this is an example of what you can see at baseline, how using the identification of a profile of proteins, you can see that the non-progressors are different to the ones that progress in pain or in structure. And using this protein profile, you can predict which patients are going to progress in the next 2 years and are going to have more pain or are going to have more structural damage in their joints. We can also see how the proteins are modulated during the progression. In the case of the non-progressor group, we see 20 proteins that differentiate visit 1 versus visit 4. In the case of the P group, 16 proteins that are different here. And in the case of the S group, 27 proteins that are different here. So this is something that we are now working with, but I show you this as an example of how can we find biomarkers, proteins that can discriminate between the different profiles, between baseline and after progression, or predict at baseline if the individual is going to progress. Another study that I will show you as an example is a study to predict if we can, to see if based on protein biomarkers, we can predict the future response of the patient to treatment. And as I told you before, really, osteoarthritis has not effective treatment, but some years ago, there was this clinical trial examining the effect of cis-ADOAs, conductyl sulfate and glucosamine, comparing them with a corticoid, celecoxib, to see if they are effective in the treatment of osteoarthritis. And so they set up this clinical trial, randomizing the patients into these two arms. One arm was the patients were given conductyl sulfate and glucosamine, and in the other arm they were given the celecoxib. And we used this cohort to analyze at baseline different protein profiles, and in the discovery phase we saw some candidates that we validate then, qualified, and then we obtained these two proteins, orosmucoid-2 and thrombospondin-1, and we insert them into predictive models using classical statistic regression models to see if using these models based on clinical variables and the proteins, we could be able to predict if the patient was going to respond to celecoxib or to droglycan, that is the combination of conductyl sulfate and glucosamine hydrochloride. So as you can see here, with an 84% of accuracy, we can classify those patients that are going to respond better to droglycan. And in this case, we developed this tool that is called Restova, that the reasoning of it is that the patient comes to the clinic, and we can measure these proteins, and then you can decide if celecoxib, to give the patient either celecoxib or either the cis-ADOA. So this is an example that can be translated into clinical practice. And then finally, as the last part of my talk, I will show you some examples of how biomarkers, the use of proteomics can be used to predict, to find biomarkers that can be used to predict the incidence of osteoarthritis. This is very important, and a lot of research focus is put here because as early as you diagnose the disease and as early as you look at this molecular rearrangement that is taking place in the joint is when you can start the early management, you can correct modifiable risk factors and restore the joint homeostasis, trying to stop and as slow as possible the progression of the disease. So it's when the interventions are more prompt to success, it's as early as possible. So some years ago, we discovered, using protein microarrays, we discovered proteins in serum that were associated with the severity of the disease. We used our cohort, the ProCoA cohort, and in a screening, we using these protein arrays where we screen like almost 200 different proteins in the serum of the patients. Then we verified this data in a novel set of patients, and we found 3 candidates, complement 3, ETH1 and S100A6, 3 proteins that could classify the OA patients from healthy controls. So in this case, they were associated with the severity of the disease. The increased presence of these 3 proteins was related with the severity of osteoarthritis. And so more recently, this is something that we published in 2021, we aim to validate this in another cohort, in this case the American cohort, the Osteoarthritis Initiative cohort, and try to see if these markers that are associated with the severity of the disease could be also useful at the beginning, more early, to predict if the disease is going to be developed. So if we can look at it early in time. So we took baseline samples from this cohort, all of them without radiographic knee osteoarthritis, so all of them were radiographically healthy. And we used 200 subjects as exploratory cohort and 133 subjects as replication cohort. As you can see here, we made models using the 3 proteins separately and also the 3 of them combined. And as you can see, CV are only the model built only with clinical variables. And as you can see here, the introduction of ITH1 is the only one that is statistically significant, that improves the accuracy of the model with statistically significance. So with the clinical variables, you get an 81%, and adding the ATH1 to the model, you raise this to an 87% of accuracy. So you improve the capacity of the model to predict which individuals are going to have osteoarthritis in the next years. So We replicate that in a second subgroup, and as you can see here in these survival curves, the people that has in their serum higher levels of ITH1 are the ones that will develop early in time osteoarthritis, knee osteoarthritis. So this is an example of how these protein biomarkers can be introduced in clinical practice. And another one, another study of a predictive model of knee-OA incidence is this one that we published a couple of months ago, in which it was based on a discovery of a panel of peptides that we detected some years ago in the serum of the patients. In this case, they were peptides. We patented this peptide profile that is built with 11 circulating peptides. And what we did is we developed an assay based on Luminex in collaboration with Merck. We developed a Luminex-based assay to measure the 4 proteins that contain these 11 peptides. So this ApoA1, ApoA4, CA2G, and A2AP. These 4 proteins contain these 11 circulating peptides, and using this multiplex assay, we screen the OAI cohort, using also separating at baseline those patients that were radiographically healthy at inclusion. So we screen these peptides, these four proteins in all these patients, around 400 patients, and we built this model. The clinical model had these traditional risk factors of OA, the age, the sex, the body mass index, and a pain scale, the WOMAC pain scale. And with the introduction of, and with this clinical model, we classify correctly those that have 70% of the patients. But when we introduce our protein biomarkers, we increase this accuracy to 83%. So you can see here how significantly you improve the prediction ability of your model by the introduction of these protein markers. And this was also replicated in our cohort, in the PROCOA cohort. In this case, we increased to an 82.82, and there were no statistically significant differences between the results obtained in one or the other cohort. So this means where there are not statistically significant differences is that the validation, that you have validated your biomarkers, your model externally into different cohorts. So with this instrument, we developed this model and build this nomogram that is called nomogram that can be translated into a calculator that the clinician can have in the consult. When the patient comes, translated the variables that are included in the model into points. So if the patient comes and it is a woman, he or she will have more points than if he is a man. Regarding the age, you can translate this here with a line here, and you have points for the sex, for the age, for the degree of pain that you have, for the weight that you have, the BMI, for the levels of these 2 biomarkers. The higher the serum A2AP, you have more points, and the lower the serum CA2G, you have more points. And then you have a total, you sum all these points. So I don't think I'm explaining it right, but this serum AP, the increased levels of this serum AP give you more risk, and decreased levels of this other serum give you more risk. So this is like a risk. This is a risk factor, and this is a protective factor, having increased levels of this protein and decreased levels of this other. And you sum all the points here, and you have a total of points that is translated into a risk of incidence. And this is important because for the patients, if you want to act on modifying risk factors, for example, an obese woman that comes to consult, and you say you are going to have osteoarthritis in the next 2 or 3 years, it's different to say that with numbers and with an algorithm that gives you points and says you have 85% of probability to have osteoarthritis in the next 2 years, then the woman will be more serious in taking, collaborating with the clinician and taking, losing weight or taking exercise and so on. So in practical, this is useful. So to correct lifestyle changes, for example. So this trying to predict what is going to happen to the person in the next. In this case, we predicted up to 8 years in advance. So these are examples of how this can be implemented. This kit, this application, this algorithm is called DITOVA. And if the patient comes to the consult, we have the serum, we measure these proteins in serum, and we have this algorithm to classify the patient. And if the patient has already NOA, we can look at the severity of this NOA. But if he doesn't have NOA, we can predict it 8 years in advance. So you are going to have this, with this percentage of probability, you are going to have any osteoarthritis in the next years. This was funded by the Galician Health Service. And finally, I go to take-home messages. These are examples that, and what I want you to share with you is that osteoarthritis is a very complex disease. It has several important unmet clinical needs, especially regarding therapeutic management. And it's time, I think it's time that we stop considering older people that they are going to have pain in their joints, are going to have movement limitations, because it is that. But we have to change that into that we can do something for that, to stop that. But for this, it's very important to implement personalized medicine strategies. And this needs to improve the characterization of the disease at the molecular level, to characterize the disease and the patient at the molecular level. Proteomics approaches, I have shown you how you can detect both in vitro and also in serum, discover and validate novel biomarkers that can be useful to implement in clinical practice. And also that these protein biomarkers can be, I have shown you some examples of how they can be combined with clinical variables, and this will enable precision medicine in a way. And that's all. And I would like to, this is my group. This is my group in La Coruña. Our hospital is near the sea, so it's very nice when it's sunny. It's not always. And this is my group in proteomics. We have a platform in the ICTS that is an infrastructure of technologies. There's a network of technologies that we have in Spain, because one limitation of doing proteomics is that the instruments are very expensive. So we work in this non-invasive platform that belongs to the CIBER BBN for bioengineering, biomaterials, and nanomedicine. So we provide proteomic services. If you want to carry out a proteomic study, you can contact us, and we have the instruments. You can send us your samples, and we can analyze them for you, and then send you the results. So it's very convenient. And also our collaborators in the different approach and in other studies. And thank you for your attention. If you have any questions.
Thank you, Cristina, for this brilliant talk. So, open, yeah.
So when you were talking about the clinical trials and trying to enrich your population such that you can see the effect of these drugs that you're trying to develop, then you have to be very careful about the results, right? Because you have selected a very specific population. How do you take that into account?
You correct it by the population. You can see when you look at the clinical variables in the study that we make on the OAI, the population was already enriched a little bit, and as you can see, the clinical variables already classified correctly as 70% of the population. So if you have a woman that is obese and that has a lot of weight, so you are enriching that in people that are more likely to progress. But then if you see that your marker statistically significant improves the classification performance, then you can translate it to other cohorts that are not so enriched.
Because in this result comes from a population that is not enriched. The AUC you're talking about.
The AUC in the OIA comes from a population that is enriched. Yes.
But it's already significant, so you can extrapolate that to another
Yes, yes, correct. Yeah, yeah. In the case of the approach, this means that in the OA there's another phenotype that is called rapid progressive. So there are people that in 2 years' time or in their joint gets completely destructed. So this is very important. And there was a clinical trial on a drug that seems to be effective, but there were some secondary effects, and it's that the proportion of rapidly progressive individuals increased with the use of this drug. So if you can characterize and predict which people are going to progress more rapidly, you will see the effect of the drug more easily. So in the case of the clinical trials, and as I told you before, the placebo effect is very important because you usually recruit to a clinical trial people that already have pain, and pain is something that can be intermittent. So you have periods of time where you have a lot of pain and periods of time that you feel better. And when you introduce the people in the clinical trials, when they have pain, a lot of pain. So this is something that has to be also considered because then you start giving the drug, and maybe the drug is not doing anything, but you see in all the clinical trials that you do in osteoarthritis, you see that the people that are taking the placebo have a reduction in pain really remarkable in the first 3 months of that you are giving them nothing, but they feel better. And this is because you have recruited them when they were in a peak of pain. You know, so this is something that, but they are statistical methods to try to correct that using these variables, and you can correct it.
I also have another question, which is maybe not too much related to this discussion, but when you introduced precision, maybe for everyone here, when you introduced precision medicine, it came to my mind because you put, how do you call this, una diana? You put.
Yes.
You put the target, and the dart was in the middle.
Yeah.
But that's being accurate, not being precise. Being precise means you shoot 3,000 times, and the 3,000 times go in the same place, even if you're not accurate. Do you think we should change the term maybe because we can be so precise at being wrong?
Yeah, you're right. You're right. Yeah.
Why don't we say like? Yeah,
it's good to be accurate, and it's good to be precise. When you develop and when you validate and analytically validate an assay, you have to be both accurate and both precise. So there's different terms, or different aspects of your assay, but you have to be good in both of them. So it's, if you are precise but not accurate at all, it's also not good. So but you're right.
But actually, that's an important point. Accuracy, precision, specificity, sensitivity. That's a take-home message. It's good to not confound the different terms because each one has its function.
Hi, thank you so much for the talk. When you were describing the different endotypes, you mentioned for the aging endotype that it can be treated with senolytics. As far as I know, there has been one clinical trial that injected senolytics into the knee and it failed. So I would like to know if there is any evidence that senolytics help with osteoarthritis.
The one has failed, but there are others, and there are other molecules that have senolytic powers that are promising for that. And in fact, one of my colleagues in La Coruña, I work in the proteomics unit, but then we have a cartilage biology unit. She's called Bea Carames, and she has some research articles looking at senolytics and xenomorphics for drug treatment, and they are now, they did a screen of drugs, of senolytic drugs, using a platform that is in the University of Santiago de Compostela, that has a platform from drug screening, and they have found 2 molecules that are going to be included now in it's a project funded by the Institute Carlos III to do a clinical trial with that. So they are promising molecules with senolytic effects that can be.
Yeah, so there is still no good, not clinical trial with a positive result, right?
No,
yeah. I wanted to make sure that I'm up to date.
No, not yet. Yes. No, you're right.
Yeah,
there are other molecules that are now being developed.
Yeah, plenty of them. Yeah, and another question: is your predictive algorithm already being used in some clinics, or?
Is a predictive model that's used now?
Yeah.
No, no.
Yeah, and when do you think it might be implemented?
Everything is moving quite quickly now. So I think in the next 5 years we will have, because there are many molecules that are being now screened. The problem is that, in the meta-analysis, the heterogeneity of the studies that we have, because, for example, the prediction of incidence that we have made is based on the inclusion of people that have both knees healthy and a KL of 0. So they are totally healthy radiographically. But when you look at other papers, they include as healthy people with KL 0 or 1, because you consider incident radiographic neostatitis from KL 2 in advance. So, but when you have KL1, you already have radiographic features. They are slight and subtle, but you can. In our case, we only consider KL0, and also that both knees are healthy. And in many cases, in many studies, traditionally they have looked at incidence and predict incidence of osteoarthritis, looking at each knee individually. So they counted knees. And this is a problem because a very important risk factor for knee osteoarthritis is contralaterality. If you have already osteoarthritis in one knee, you will be more prone to have osteoarthritis in the other one. So when you trying to do meta-analysis with this is very difficult because there is a lot of heterogeneity between the different studies, the inclusion criteria, the outcome, etc. So you have to be very careful with that.
Yeah, thank you so much.
No, you're welcome.
Thank you so much for the talk. I think that you have been a really good overview of the current state of osteoarthritis. And from the different biomarkers, that there are a lot of them that you have presented, have you looked if they are affecting the different phenotypes of osteoarthritis, or they are like an overall biomarker of osteoarthritis?
I haven't looked at that. So they are explaining different things. So some of them are looking at cartilage degradation, but this cartilage degradation can be common to the different phenotypes. So if you want to look at characteristics that mean more endotyping, this is something that has not really been yet well investigated. So the theory of endotypes and phenotypes is from the last like 4 or 5 years, more or less. So now more and more are using machine learning tools and using data-driven hypotheses. They are trying to combine all the data and trying to characterize better the endotypes. But all the work that was done previously with all these CTX2 and all these markers that have been classically, they only explain what is the collagen degradation, for example, and this has not been associated with a specific phenotype or endotype in particular. Mm-hmm.
And do you think that from serum samples we could get this phenotyping?
If you can, the problem with serum is, of course, that being circulating, you can look things that could come from very different aspects. What we have done is we have made a lot of studies trying to correlate what we see in serum and what we see at the level of synovial fluids, for example. And in many cases, they don't correlate at all. So, and also, if you look at the tissue and then at the synovial fluid, they don't correlate either. They are proteins that are decreased in the cartilage and then increased in the synovial fluid. So it's something that you have to look at the point. The advantage of serum is that what you are seeing there is something that will be very easily applicable in clinical. So we have seen these markers that in the last predictive model, they are very traditional. Apolipoproteins are proteins of lipid transport, so they are not, how can you correlate them with the pathogenesis or the physiological things that are occurring in the joint? Maybe they are not directly connected, but if you see that, and it's something that is robust, and you see it, you can confirm it with accuracy, sensitivity, and this gives you a good predictive value, positive and negative predictive value. So it's something that you are looking, and then you can study where could be the thing. from the 4 markers that we analyzed, we took out the ApoE4 because it didn't show good results. We kept with 3, and using only 2, that are the CA2G and the A2AP. We only looking at these 2 biomarkers is where you have the best performance. And one of these is a protease, is a protease that is very typical in serum. So you can connect that with the biology of what can be happening, but of course it's something that is circulating, so it doesn't have to be related with the gene.
Okay, thank you so much.
Yep.
Thanks for the presentation. Sorry, I'm not sure it's related to your topic, but if a classifier is giving a precision recall AUC values of 1, so how would I interpret that?
Sorry, I didn't understand.
A classifier
Yes?
giving a results of precision recall and AUC curves of 1, suppose.
Yes.
Then how would I interpret that classifier?
A classifier with a precision of 1.
Precision 1, recall 1, and
Yeah.
AUC values are 1.
That is perfect, no?
Yes.
Yes. And
So I feel like it's working well, or
yeah, it's working well, no?
In general, machine learning people start to have serious doubts when the value is 1.
Yes.
I think this is for the risk of overfitting.
Overfitting, yeah.
So, but I guess that so basically then when you have the result of a classifier, so you go through verification and validation,
Yes,
no?
yes.
Independent validation.
Yes, we usually when we do in a discovery cohort, if we have an N sufficient big size of the group, we do an internal cross-validation in the first cohort, and we try to extrapolate. In the case of the second one, we developed the algorithm in the OAI cohort, and we validate it externally in the ProCoac cohort using the same metrics that we developed in the first one. We extrapolated to the other one, to the second one, and if there were overfitting in the first one, then we would see that in the external validation. And so it's obviously it behaves worse in the validation, but if it behaves good enough and you don't see statistical differences between the two of them, is when we consider it validated. I think we are.
Yeah, and
I'm not a statistic.
and in terms of clinical application, so below which accuracy or discrimination value would you deem that the results are becoming?
Well, it's said that more or less 80%. It's a very good biomarker. If you have more, there have been works published with 70% that claim that it's a good marker. If you get to 80%, is really good. Then you have to look also to the predictive values, because osteoarthritis is a very prevalent disease. So the predictive values are the specificity and sensitivity are dependent also on the prevalence. And if you go to the predictive, so it depends on the biomarker, the use do you want to do in clinical to a biomarker, if you want to put it into a screening procedure, or if you want to do it for guide decision making. In this case, this is a screening because if you know that people are going to have osteoarthritis in the future, you will modify lifestyle changes or so on. It's not a critical decision that surgery depend on that, for example. So it's dependent on the objective of your model, that you want to have higher one thing or the other. So.
So I don't know, I missed your first starting slides.
Yeah.
So it's dataset name of the dataset.
The name of the dataset.
Yes.
The name of the dataset it's a cohort.
There are three main cohorts. ProCoac cohort that is in Spain. There is an approach cohort that is an international consortium, and there is
an American
Osteoarthritis initiative, which is a public US-based cohort.
So it's a public or private?
The NIH, the Osteoarthritis Initiative, is public. Yeah, you can ask for. They have run out of samples at baseline, for example, because everyone asked for samples of baseline, and also they have a small amount of non-exposed, what they are called non-exposed, that they are really healthy people. They were like 130 or 150, and so they were also very run out of that. But if you want data, you can ask, and you can retrieve even MRI images, X-ray images, and what's good is that it has a long follow-up. So it's like more than, I think, 10 or 12 years follow-up. So for osteoarthritis, that is a very slow progressive disease in general, it's a good cohort to study.
So I think we have another question here around.
Sorry.
Yeah, all right.
Sorry.
Sorry. I have just two really short questions. The first one is regarding the model you used for the classification, because you showed many rock curves, but I really wonder what is the underlying model, like the algorithm, is it like logistic regression or
multivariate logistic regression, yes. It's traditional statistics, yes. We first do an univariate, we show univariate models to see the variables that are more contributing to the classification, and then we integrate them into a regression model.
Okay, but this is like the normal model you use, or you also tried other models and then compared the rock values, like the AUC, or you just focused on this linear logistics?
In this case, we focus on that, on the linear one, yeah.
Okay, and the second really short question is about the scale you showed. I think it was like the really last application you showed for the, I think it was to predict the progression of the disease.
Yes.
Then we have like this many biomarkers or like features like
the nomogram
and so on. Exactly, and then you have like this scale, and I just really wonder how you define, like how important which feature is and how to.
It depends on the weight that each variable has to the model. Yes, I really didn't develop it. The statistical people in the hospital were the ones developing it. So sorry, I cannot go. So I know that there is a mathematical expression of the model, and the coefficients that are multiplying each variable are the ones that are putting into the graphic to build the nomogram. So it depends on that.
All right, I've never seen it before, but I think it adds really a lot of interpretability, also for the patients to see what kind of lifestyle changes cause which kind of changes in the prediction and progression of the disease.
I mean, this is a way to put it in mathematical, in an expression, in numeric degree. Everything is somehow interpretable, because the pain is something that is interpretable. Even the radiographies are something that is interpretable. You can see the graduation of the XL usually needs to be made from 3 different observers, because many, depending on the clinician that is looking at the radiography, they will give a different grade. So we put, one of the variables that were put in the nomogram was a pain scale. So that is something that also will depend on how the patient feels. And for example, in the case of the approach cohort, that it's this one that 4 countries, there were 2 centers in the Netherlands, one in Oslo, one in Paris, and our cohort in Spain. And it was really remarkable to see that the progression in pain was significantly higher in Spain than in the other centers, and that's why. So Galician women are complaining more about pain, so it's something that you have then to correct by center, the results that you have. So everything is kind of interpretable, but then there are mathematical tools to try to correct that and to try to put it as objective as possible. That is my feeling.
Exactly. What I just meant is it's nice to see as a patient what effect does it have if I change my lifestyle, if I move more, if I change my diet, what would be the progression? Like
it's
really
something that they with the rheumatologist is a person that, so rheumatology diseases are chronic diseases more, so they have should have like a good collaboration with the physicians. So patients and the physicians are going to be together for long time because they are chronic diseases, so it's good that something, and in this case, that's why we are talking about precision medicine and not personalized. Personalized means more like a drug thing, so I'm going to give you a drug that's personalized for you. In the case of precision medicine, we are moving more for the, you know what precision medicine means, what all the P's, and one P is participative, so that the patient participates with the clinician into the management of his own disease, so it's something that we need to do.
All right, thank you.
You're welcome.
Yeah, I think in this discussion, correct me if I'm wrong, but I think in this discussion, so related to that question, it's important to make the difference between the stratification you want to make on patients that already have pain. So here, so basically then the different dimensions of pain, including sensitization, are very important, and it makes probably much more complicated the identification of biochemical biomarkers. And then, so the exercise that Cristina is doing to try then to predict, so starting, so with a longitudinal follow-up starting from people who still haven't developed osteoarthritis. So here pain is much less important. But yeah. So
for the FDA, both things, and for the EMA, both aspects in osteoarthritis are important. And there was one drug that was tested some years ago that seemed to really stop the structural damage, but it didn't have any effect on pain. And it failed because of that. So it is not only that you work on the structure, but you have only to modify the pain and try to lower the pain. So it's something that is complicated. That's why it's so challenging. But hopefully in the next years we will have, because there are many molecules that are being screened now. So
a bit related to that. So basically, if pain is an important symptom that you take into account and to evaluate then the trajectory of your patients. So do you have in any cohort an evaluation of the sensitization?
No. It's all for my clinical assessment. So this is questionnaires and pain scaling. This WOMAC pain scale, in the case of the approach, they use another scale that is called COOS, but they are pain scales that are based on clinical assessment by the physician, and you rely on questionnaires, but not sensitization.
So what do you think you could use as an objective biomarker of the real state of osteoarthritis once you have symptoms? What would it be then, the Kellgren-Lawrence? Would it be the objective one? What do you think?
Objective, in the case Kellgren-Lawrence is an objective measurement by radiography. Both things, the estimation of pain and also radiography. In fact, they are now with this massive trying to, because as osteoarthritis is such prevalent, studies should be, the limitation that approach had is the limited number of patients that could be recruited, because with only 300 patients, you can, and only 2 years of follow-up, the results of this cohort have been rather limited. And in the last years, there have been a lot of studies with data from, for example, from the UK Biobank, because the UK Biobank, as you know, it's a cohort that has a lot of data from primary healthcare and so on. But in the case of the studies that have been performed for osteoarthritis using data from the UK Biobank, the problem is that only with pain, they assume the person has osteoarthritis, that there is no image assessment because the patients do not have radiography. So the results from there have to be taken with caution, because there will be, there is a lot of background noise coming from a wrong diagnosis, that you take somebody that comes and says my knee aches. Oh, okay, you have osteoarthritis, and probably the radiography is completely healthy, and it's idiopathic thing or something. So it's something that, yeah, there is a question there.
And as well, the other side, there's going to be plenty of people that has structural damage on the knee and they have no pain.
Yeah.
That's also very important to take into account.
Yes, yeah, totally. Do not correlate. It's something that are two aspects of the disease, but the one doesn't mean the other. So yes, and even they are drugs that are aimed to lower the pain than provoke more damage in the structural damage, because the person doesn't feel any pain and it goes on and the joint goes worse. So something. Mm-hmm.
So maybe a last question, if there is no more questions from my side, is so among the different molecules that the proteomics that you're identifying, some of them would be associated with fast biological processes, some other would be associated with slow biological processes. So how easy can you make the difference, and how does it impact on the identification of a really exploitable biomarker?
How can be? Well, I don't know. It's a very tricky question. How can they be associated with slow or fast?
Yeah, because fast processes will be high dynamics,
yes,
for example, and then slow processes would have a stronger inertia.
Yes.
So might be maybe more robust than in order to interpret over several years of evolution than the fast processes.
Yeah. Well, I don't know really.
Because I've seen that you make a network representation of the knowledge that you have.
Yeah.
So how is the interpretation of this network then useful in order to evaluate this kind of problems or these kind of questions?
These kind of questions. I don't know. So it's kind of so yes, once you have the picture of how it's evolving in the individual, so we try to identify markers that are robust, that are easily detectable, that are not fluctuable due to different situations, and we think that looking at those would be the most applicable then in clinic, the most easily translatable in clinic. This does not mean that there are not others that can be like more, detecting, for example, inflammatory phenotype in some stages. But osteoarthritis is not really a disease, because we are studying other diseases such as, for example, rheumatoid arthritis that are like more flare dependent. So the flares are much more striking than in osteoarthritis. You have pain periods with more intensive pain, but they are not such as in other, like more heterogeneous diseases like other rheumatic diseases such as osteoarthritis. So trying to find just robust things, and we looked at the work that we in the pain detectives that we shared. We talk about another work in which we analyzed autoantibodies in osteoarthritis and how these autoantibodies could be also putative good biomarkers of the disease, and this is something that has not been explored at all, because osteoarthritis is not an autoimmune disease like rheumatoid arthritis, for example, but we did detect some autoantibodies that could be synthesized, and these autoantibodies are known to be like more stable also than proteins that have a quicker turnover, so that they are, and we select some of those, but this is something that has not been then implemented because they are more difficult to analyze than in clinical practice. It's much easier to quantify a protein then.
Yes, thanks.
Sorry.
Okay, so if there is not much questions, so
thank you.
Let's thank again Cristina.
Thank you.